So when the blood pressure goes down, the perfusion, renal perfusion reduces, and the Pulcine cell releases the renin, and macular densa also releases the renin, and then it comes into general circulation. Angiotensinogen, which is available already in the circulation, is converted into A1 in the presence of renin. A1 is the decapeptide. Already we discussed. A1 is a decapeptide. Deca means 10 amino acids. It's an enzyme with 10 amino acids, right? Which is an inactive protein, right? And when A1 come into circulation, it goes to the lung, and in the alveolar capillary membrane, angiotensin converting enzyme, it convert A1 into A2. And this is an octapeptide, right? Octapeptide means it consists of 8 amino acids, which is an active protein. So what does angiotensin converting enzyme does? Angiotensin converting enzyme, it converts inactive protein to active protein. Decapeptide enzyme to octapeptide enzyme. That is, it is removing 2 enzymes. 10 enzymes, 2 enzymes it is removing, and now the A2 is present in the circulation. So we were discussing last class what is the function of A2 enzyme, right? So A2 enzymes, there are 5 to 6 functions are there. I think last class 3 functions we have discussed. First is angiotensin 2 enzyme. It has a receptor in the veins and arteries, right? So it has the veins, it has the receptor. So I'll make it here. So this is the, imagine this is the receptors present in the venous side for what? For A2 enzyme. Now A2 is circulating throughout the body. Angiotensin 2 is circulating throughout the body, right? So when it binds with the receptor in the venous side, it constricts the veins. What will happen if veins are constricted? Venous return increases, and cardiac output increases, and blood pressure increases. Which blood pressure? Systolic blood pressure will increase. When venous return is increased, cardiac output is increased, systolic blood pressure is increased. And it is having, second is, it is also having the receptors in the artery also, right? When A2 bind with the receptors of the artery, arteries will constrict, total peripheral resistance will increase, right? When total peripheral resistance is increasing, which pressure will increase? Diastolic blood pressure will increase. So this is the first attempt to increase the venoconstriction and arterioconstriction. This is the function of the A2, angiotensin 2, right? This is first function. Second is, you know that A2 is circulating throughout the body, and it will reach the central nervous system also. It will go to the central nervous system also. That enzyme, A2 enzyme, will go central nervous system. In central nervous system, it will reach hypothalamus, right? You know where is hypothalamus? You don't know? It will reach the hypothalamus. Hypothalamus is also having the receptor for A2, right? Hypothalamus में जाके वो क्या करेगा? It will, in hypothalamus there is a center for thirst. Center for thirst, है ना? You know what is thirst? Yeah. Thirst के लिए there is center होता है. When A2 go and bind with the receptor in the hypothalamus, the patient will get thirsty, and he will ask for water, right? So water, if he is drinking more and more water, then what will happen? This water will go and add to the blood, right? And the blood volume will go slightly up, right? When blood volume is going up, then what will happen? Venous return will increase, cardiac output will increase, blood pressure will increase. So this is second attempt by the A2 to increase the blood pressure. We are discussing initially the blood pressure gone down. So our body is trying to increase the blood pressure. First is it is trying to constrict the arteries and veins and increase the blood pressure, right? Second step is it is trying to stimulate the hypothalamus, and then it increases the thirst, and then it is trying to increase the blood volume, which in turn increasing the venous return, cardiac output, systolic blood pressure is increased. This is second attempt. And third attempt is the body is trying to see all the sympathetic nervous system, all the sympathetic nervous system, nerve endings, sympathetic nerve ending has receptor for A2. Sympathetic nerve ending, suppose imagine this is a sympathetic nerve ending, it is having the receptor for A2. When the A2 angiotensinogen 2, when it binds with the sympathetic nerve endings, then the sympathetic nerve ending will release norepinephrine. Norepinephrine will be released by the sympathetic nerve ending. When norepinephrine is released in the from the sympathetic nerve ending, this has three functions. Right? Norepinephrine also constrict the veins, right? And it also constricts artery, right? It constricts the artery. So norepinephrine induced venoconstriction. First is A2 itself directly constricting the veins and directly constricting the artery. That is a direct effect of A2. Now because of A2, it binds with the sympathetic nerve ending and norepinephrine is released, and norepinephrine is also having effect on veins and arteries, and it constricts the veins and arteries. समझ में आया? So previously A2 induced venoconstriction. A2 के वजह से venoconstriction हुआ. Now norepinephrine induced venoconstriction. Again arteries and veins are constricting and trying to increase the blood pressure. Right? And third point, norepinephrine, it is having a directly stimulatory effect on the SA node and AV node. When norepinephrine is released, SA node and AV node is stimulated. And what happens if SA node, AV node is stimulated? What will happen? Heartbeat will increase. Heart rate will increase. If heart rate is increased, cardiac output is increased, systolic blood pressure will increase. So this is third attempt of A2 to increase the blood pressure. So from A2, from where A2 came? A2 came from A1 in the presence of ACE, and A1 came from aldosterone, right, in the presence of renin, and renin came because of release of, because pressure is low, this juxtaglomerular apparatus has released the renin. Are you clear what I am saying?
Yes.
So renin came, फिर उसके बाद angiotensin 1 came, फिर angiotensin 2 came. Now we are discussing the function of A2. First function, venoconstriction, arterioconstriction. Second, hypothalamus is stimulated and thirst is induced. Third is norepinephrine is released. Norepinephrine is released, and norepinephrine is having three uses: venoconstriction, arterioconstriction, cardiac stimulant, cardiostimulant. So three functions we have discussed. Next is fourth function of A2, angiotensin 2. So now it will act on adrenal gland. Act on adrenal gland. You know adrenal gland, there are cortex and medulla. Medulla and cortex, right? Cortex is having, we have discussed, three it's like a flower, right? Three layers. What are the three layers? This is medulla, innermost layer is medulla and outermost is cortex. Cortex is having three layers. What are the layers that we discussed? This is innermost, this is adrenal gland. Adrenal gland innermost layer is cortex, medulla and outer is cortex. Cortex is having three layers. What is this layer called? That we discussed. Zona glomerulosa, zona fasciculata, zona reticularis. You have not come across this word?
No, sir.
Okay. So this layer is zona glomerulosa, right? Zona, Z-O-N-A, zona glomerulosa, right? And this layer is zona fasciculata, right? And this is zona reticularis, right? So zona glomerulosa, it is having the receptor for A2, right? Now you see what is circulating now. What is circulating? A2 is circulating, right? That A2 will enter into the renal artery, and you know this is the gland, right? And it will go and stimulate the what? Which layer I have told? This is there are three layers I have told here. This is zona. This is zona glomerulosa, zona reticularis, zona fasciculata, zona reticularis. Three layers are there. A2 will reach adrenal gland, and it will stimulate the zona glomerulosa, right? Now zona glomerulosa will release aldosterone. Right? Zona glomerulosa will release aldosterone in the presence of A2. Aldosterone कहाँ से आया? A2 से आया, right? A2, zona glomerulosa is having the receptor for A2. A2 will bind with zona receptor in the zona glomerulosa, and aldosterone is released in the blood circulation. Now what is happening? Aldosterone is now circulating. I'll give another color, green color, right? So this is aldosterone. Now aldosterone is circulating throughout the body. Are you clear what I am saying? In the beginning, what was circulating? Nine. Before that, before blood pressure was angiotensinogen, which was secreted by the liver, is circulating throughout the body. When renin came into the blood, angiotensinogen converted into A1 in the venous system. Right? In the beginning, venous system, it was A1. When A1 went to the lung, it is converted into A2. Now A2 is circulating throughout the body. And when A2 is circulating, meantime what it will do? It will constrict the artery, it will constrict the vein, it will stimulate the hypothalamus, it will stimulate the sympathetic nervous system. All these will happen, right? And A2, when it goes to kidney, when it goes to when it goes to adrenal medulla, then what will happen? It will stimulate the adrenal medulla, and then zona glomerulosa will release aldosterone. Now aldosterone came into the circulation. Now aldosterone is circulating throughout the body. Are you clear?
Yes.
First angiotensinogen, right?
Yeah.
फिर angiotensin 1, angiotensin 2, Then now aldosterone. Now aldosterone is circulating in the body. Now we have to study what is the function of aldosterone. Aldosterone is also having some effect in body, in cardiovascular system to increase, right? So what is aldosterone does? Now aldosterone function we will see how aldosterone it works. Okay, this aldosterone it works on the special cells, that is primary cells and the collecting tubule. Now A2 is circulating, right? So A2, when A2 is coming here, right, it acts on the primary cell near the collecting tubule. This is collecting tubule, right? Nephron ka collecting tubule. So this is called, here the cells are present which is called primary cells, right? And this A2 acts on primary cells and it acts on proximal convoluted tubule. A2, see, this is primary cells which is near the collecting tubule, and these are the cells which are nearby the proximal convoluted tubule. This is proximal convoluted tubule, and this is loop of Henle, and this is distal convoluted tubule, and this is collecting tubule, right? So A2 is having receptor, right? Sorry, aldosterone. Now aldosterone is in circulation, right? Aldosterone, it is having a receptor in the primary cells of collecting tubule and the cells of proximal convoluted tubule, and it helps in reabsorption of sodium and bicarb. It helps in reabsorption of sodium and bicarb. I will tell how it works, right? So I will draw one proximal convoluted tubule. This is collecting tubule, and I have drawn one right. And what is this? Afferent arteriole and efferent arteriole, right? So now, what is circulating in the body? Aldosterone. Aldosterone is circulating in the body, right? So when aldosterone, it enters into the primary cells near the collecting tubule, so it activates some genes. It activates some genes and it synthesizes three types of cells. One is so aldosterone enter into the primary cells and it combines with the receptor, and this will stimulate the gene and synthesize new types of cells. समझ में आएगा? A2 it enters into the aldosterone, it enters into the primary cells and it combines with the receptor and it activates the gene and synthesize new cells. First is this is the sodium potassium ATPases. ATPases are synthesized. Sodium potassium ATPases are synthesized from the cell and it is planted in the walls of the primary cells. Right? Sodium, this is sodium, potassium, ATPase. ATPases are synthesized. For that, what is required? Aldosterone is required. Aldosterone went into the primary cells, activated the gene, new cells are produced. Three types of cells are produced. First is sodium potassium ATPases, right? Sodium potassium ATPases are synthesized and they are planted on the walls of the primary cells. So what is the function of sodium potassium ATPases? It will throw the sodium outside the cell. Sodium which is present inside the cell will be thrown out. That is the function of the sodium potassium ATPases, right? So what will happen here? The sodium level will become low. Normally intracellular sodium is high. सेल के अंदर सोडियम ज्यादा है क्योंकि यहाँ पे ATPases आ गया है तो सोडियम बाहर चले जाएगा. It will go out and the sodium level will become less in the primary cells near the collecting tubules. यहाँ तक clear है? फिर second what it activated, second gene, it activated the gene and it synthesizes the sodium channels. Sodium channels, that is gateway for sodium. And now this sodium channel is synthesized, new sodium channels are synthesized and they are planted in the walls of the collecting tubule. These are the sodium channels. More and more sodium channels are created and planted in the collecting tubule. Previously it was walls of the primary cells. Now it is placed in the collecting tube, right? Okay, when sodium, already we discussed, sodium is entering here, 65% of sodium is absorbed here, and here 25% is absorbed sodium here, and here 10% is available. It will be losing in the acid. 10% sodium will normally go out in the urine, right? And now what happened, because pressure is low, right? This is less than 65, this is greater than 65 because it get more time, we discussed. Yes. Because fluid is moving slowly, more and more sodium is absorbed here, and here also more and more sodium absorbed, and here will be less than 10%, and that is the reason why the macular densa is stimulated, right? Now sodium is coming here, but less sodium is coming. समझ में आ रहा है क्या? Sodium is coming, but less, right? And that sodium will also try to enter into the cell. Now because here the channels, new channels has been created, right? So already sodium has gone out. Because of sodium potassium ATPases, sodium has gone out and this is less in sodium. So the sodium which is coming here will try to enter the primary cells. Are you clear? And third is, one more gene is one more cells are synthesized, and that cells are potassium channels. Potassium channels are synthesized and planted in the collecting walls of the collecting tubule, right? Okay. Now the sodium will enter here. In exchange, potassium will go out. Potassium will enter the collecting tubule. Are you clear what I am saying? So first is sodium potassium ATPase. Sodium potassium ATPase is common channel for both sodium and potassium. Sodium has gone out, potassium will come in, right? Sodium has gone out, potassium has come in. Sodium is now inside the cell, sodium is less and potassium is high inside the cell. Now that potassium will go out, and because sodium is less, sodium will enter here, right? In exchange for sodium, potassium will go out. So now the potassium will, you can see the potassium in the urine. More concentration of normally sodium, but now the sodium, all the sodium has been reabsorbed. And one thing you should remember, along with sodium, water will also move. Already we discussed. Wherever the sodium goes, water will follow, right? So water also will follow, sodium will enter, and this sodium will enter in. Sodium and water will enter into the blood circulation. And what happens finally? The blood volume will increase, urine output will reduce. Urine output will reduce because sodium is going out, water will also go out, and this water, sodium and water will mix with the blood and it will add up in the blood volume, and venous return is increased, cardiac output is increased, and blood pressure is increased. Are you clear? So A2, it bind with adrenal cortex. Again I am repeating. A2 bind with adrenal cortex, and adrenal cortex release. Adrenal cortex mein which layer? Zona glomerulosa is stimulated. Aldosterone is released. Aldosterone bind with the primary cells because it is having receptor. It binds with the primary cells, and then it activated the gene, and three types of cells are produced, synthesized. Sodium potassium ATPases. Sodium potassium ATPases is planted in the walls. Sodium potassium ATPases means it's two-way channel. Sodium will go out, potassium will come in. Sodium has gone out, so the level of sodium, intracellular sodium is less. When sodium is coming, it will enter into the cell because here new sodium channel is created. So sodium will enter here. Because sodium is entering, in exchange potassium will go out. This potassium from where it came? It came from outside. First it entered the cell, primary cell, and from primary cell it entered to the collecting tubule. So now the potassium is leaking in the urine instead of sodium. Because already sodium is absorbed here, here and here and here, still more sodium is absorbed. Why more sodium is absorbed? Because water will follow sodium. And water is required to increase the blood volume, right? And blood volume should be increased to increase the cardiac output and systolic blood pressure. Any doubt? So this is the function of aldosterone. Aldosterone, it has two functions. One is it acts on the primary cells, and what second I have told? It acts on the cells of proximal convoluted tubule. So this half I had completed. Second half is it acts on the proximal convoluted tubule. Now I am taking one cell near the proximal convoluted tubule. What is the function of? See, sodium and bicarb HCO3 is filtered. Sodium and bicarb is normally reabsorbed, right? Sodium will enter into the cell, and this bicarb HCO3, see, when sodium is going out, hydrogen proton will enter into the proximal convoluted tubule. Again I will repeat. Sodium is, I have told 65% of the sodium is reabsorbed in the proximal convoluted tubule, right? So when sodium is going out, in exchange, proton will enter into the proximal convoluted tubule. And here what is filtered? Bicarb is filtered, right? So H plus will combine with HCO3 to form H2CO3. And HCO3 it dissociates into carbon dioxide and water. Carbonic acid dissociate into carbon dioxide and water. This water will go here as a urine, and this carbon dioxide will enter into the cell. Because already we discussed carbon dioxide, it can enter into the cell anywhere, right? Carbon dioxide will enter into the cell. This carbon dioxide, it mixes with water in the cell to form H2CO3, and this again dissociate into H plus and HCO3 minus. This HCO3 will come outside and this hydrogen ion will go inside. Right? So what happened? The filtered bicarb is reabsorbed by the proximal convoluted tubule. So sodium and bicarb is reabsorption is increased. You want me to explain again this? See, sodium and bicarb is filtered, right? Sodium will enter into the cell in exchange of hydrogen ion. This hydrogen ion kahan se aaya? From where it came? Now I will tell. This bicarb, it combined with this hydrogen ion to form bicarbonic H2CO3, right? Carbonic acid, and it dissociates into carbon dioxide and water. Water has gone out. Carbon dioxide enter into the cell, right? And this carbon dioxide, it combines with intracellular water to form H2CO3. And this H2CO3 dissociates into hydrogen ion and bicarb ion. This hydrogen ion enter in place of sodium, in exchange of sodium. And this bicarb is now coming into the cell and it will enter into the circulation. Right? Any doubt? So this is the function of aldosterone. Basically, when the blood pressure, in simple, if you want to say, when blood pressure goes down, renin is released. Renin is the main cause for stimulation of A1, A2, aldosterone. A1 is an inactive protein. It does not do any work. But when A1 is converted into A2, A2 is having many functions which tries to increase the blood pressure. Because of A2, aldosterone is released, and aldosterone is also helping to absorb more and more sodium and bicarb from the kidney, that from this proximal convoluted tubule and collecting tubule. It try to absorb more and more sodium and bicarb so that water can follow and it will add into the blood circulation, and it will try to increase the venous return, cardiac output and systolic blood pressure. Any